Well, aloha everyone, and welcome back to Human Biology here at Chaminade University. Today we are going to start Module 2, and we're going to jump right into Module 2A when we talk about cell structures and functions. And I'd be remiss if I didn't start this chapter off discussing cell theory. Cell theory states several things. First, that all life, or anything that we consider to be alive, any living organisms, are going to be comprised of cells. And that the flip side of that, the cell is the basic unit of life for all living organisms. And again, it's the smallest unit that we can consider to be alive. And when we say alive, that means that it can survive independently. All cells are going to come from pre-existing cells. So cells are going to give rise to the next generation of cells, i.e. the parental cell line is going to give rise to the daughter cells. And they do this by passing on their genetic material, or DNA, from one generation to the next via a process known as cell division. Now, cells are divided into two major categories. Cells can be either considered either prokaryotic or eukaryotic. Prokaryotic cells are much more primitive. They are smaller. They are unicellular organisms, things like bacteria and yeast. They do not have organelles or nuclei. Their DNA is going to be in the exact same area as all the rest of the cell material, whereas in eukaryotes, the DNA is going to be isolated in the nucleus, separate from all the organelles, which each have their own specific function within the cell. And we'll get into that as we move on through this lecture. As we're discussing human biology for the rest of this semester, we're going to be focusing mainly on the eukaryotic cells. And let's talk about human cells for a little bit. So human cells range in size. They're approximately 5 to 20 micrometers in diameter. The smallest one is going to be the blood cell, which is going to have to be able to travel through the capillaries to be able to deliver oxygen to the tissues and pick up carbon dioxide. Some of the longer ones are going to include things like neurons, which are going to run all the way from your brain down to your tippy toes. So some of these have specialty modifications that allow them to increase surface area. Like neurons can be very, very long and very, very thin. And then we have other types of different features that cells might have that might be unique from other cells. Examples of that include microvilli, which are little projections that come off of cells lining the digestive tract that allow the absorption of nutrients as the food passes across them. However, cells have a limit on how large they can get. So at some point, once the cells reach a certain size, they have to divide and become another cell. So in order for an organism to grow larger, they're going to increase in cell number rather than in cell size. Okay, the female egg cell is going to be the largest cell type. Here's a female egg cell compared to a bunch of the sperm cells that are attempting to penetrate here. Sperm cells are going to be the smallest cells. They are going to have all of their DNA condensed down into their nuclei here, and then they've lost all of their organelles and cytoplasm. So they're literally just little nuclei. Let's see. We also have, here's an example of microvilli and here in the intestinal lining and a neuron, which is going to have specialty regions here called dendrites that are going to get information in, and then a long axon that's going to send information out. So these are just demonstrating that there are specialty features that certain cells have that are different from other cells that allow them to have greater surface area to be able to extend long distances or have more surface area for absorption, for example. Okay, but no matter what, whatever type of cell we're talking about, it's going to have a plasma membrane that surrounds it that's going to separate the inside of the cell from the outside of the cell. It's going to be what we call semi-permeable. That means that it allows certain things in and keeps other things out. So it's a barrier that's going to be semi-permeable that's going to allow things to come in that it needs to come in and send out waste products that it does not want to come in. And we're going to maintain a gradient where we're going to have difference of what's the concentration of different materials in the outside of the cell and the inside of the cell. We're going to have fluid on either side. So the cytoplasm is going to be the fluid that's on the inside of the cell. And that's also going to have the organelles and other liquid, other solid matter inside. And if we're looking at just the fluid compartment inside the cell, that's going to be called the cytosol. The fluid that's on the outside of the cell is called the extracellular fluid, and that can be called the interstitial fluid. The blood supply also counts as this type of fluid. So basically, you're either going to have fluid that's inside the cell, fluid that's outside of the cell, fluid that's in the blood supply, and there's a constant exchange of nutrients and materials from the blood to the interstitial fluid, from the interstitial fluid to the inside of the cell. And again, the plasma membrane is going to regulate what comes in and what comes out. So the plasma membrane is really unique because it's going to be comprised of a bunch of what we call phospholipid molecules. So it's a phospholipid bilayer, and these phospholipids are able to kind of move about along the plasma membrane wherever they choose. So it's called a fluid mosaic model because it's not stagnant. It's a mosaic of these the phospholipids as well as proteins that are embedded in there. There's cholesterol embedded in there. And it's got basically a fluid consistency like an oil, which is what's going to keep the aqueous solution, right, the water-loving solution from the outside, separate from the water-loving solution on the inside, right, because oil and water don't mix. Okay, so this is what I was talking about. This here is a phospholipid. Phospholipids are going to be comprised of one region that is water-loving, it's hydrophilic, it's this head here, and then one region that is water-fearing or hydrophobic, it's oil-loving, as you might imagine. And if we align these in a long series of these, picture these like balloons on the ceiling, then we have these aligned such that all their hydrophilic heads are in one direction and their tails are oriented in the other. And then we also make another layer on the flip side where we have the exact same thing going on, where we have the hydrophilic heads in one direction and the hydrophilic tails headed towards the other tails. Now we're going to have this area where we have water-loving, water-fearing, water-fearing, water-loving. So you can see how that would be a barrier, right? So again, hydrophilic is going to mean soluble in water or water-loving. Hydrophobic is going to be insoluble in water, like these tails here. And that's going to form a nice barrier layer between the inside of the cell and the outside of the cell. Okay, but it's not just these phospholipids. Again, we have several things embedded here. Here's cholesterol. We're also going to have a bunch of different proteins. We have proteins that allow things through, like this channel protein. We have proteins that are going to be recognition proteins that state, hey, for example, these are going to be immunoproteins that say, hi, I am Dr. Griffiths' cell. I'm Dr. Griffiths' heart cell, right? Very specific. They're going to be self versus non-self identification markers. Right? So this is going to be a series of phospholipids that form the barrier that also are going to have channels whereby things can come in and out. So this is going to give us selective permeability to allow us to bring in what we want and keep out what we don't want. So inside this series of membrane, we're going to have multiple different types of proteins embedded in here. The different types of membrane proteins are going to fall into multiple classes. Major classes are going to include receptors, antigens, enzymes, transport proteins, adhesion proteins, and gap junctions, or communication proteins. So let's start with the first one. So receptors are going to be proteins that bind to the outside of the cell and somehow affect the function of that cell. So they're going to bind to what's called a ligand, which is going to be on the outside of the cell, and then send some information internally that affects the function of the cell. Some good examples of this are going to be hormones. So circulating hormones in the blood can bind to the cells and up or down regulate different cellular processes. We also have proteins that are called antigen proteins, and those are the ones that I was just talking about that are like recognition markers. These are membrane proteins that act as markers, like blood type proteins, for example. So depending on what your blood type is, you will have either A glycoprotein on the surface of your cell, B glycoprotein on the surface of your cell, both or neither, right? And those are going to be called blood antigens. And we'll talk about that when we get to blood typing later on in the semester. We'll actually be doing that in lab. Okay, so we also have enzymes. Enzymes are specialty proteins that catalyze chemical reactions. They speed up the rate at which a reaction would occur. So not a reaction that would never occur. This is a reaction that would occur normally by its own processes, but at a very low rate. And enzymes basically are going to force two different molecules together to increase the rate of that reaction. They, like matchmakers, remain the way that someone who got two people together in a couple, they get married. The person who got them together as the matchmaker remains unmarried. The enzyme at the end of this process is going to remain unchanged, so then it's able to convert the next substrates into products over and over. So enzymes are going to speed up the rate of chemical reactions by bringing together two things that are going to interact and then create the products. We also have transport proteins. Transport proteins, we have multiple different types, are going to allow substances to go across the membrane. We have channels, we have carrier proteins. They're going to be very selective to the substances that they allow through, such as like aquaporins, for example, are going to be a channel that allows water specifically through. Adhesion proteins are going to be the type of proteins that, as you might imagine, act like glue or like zippers. They're going to connect cells together. We also have gap junctions that allow communication amongst cells. All right. So we kind of touched on this earlier, but this is a brief description of the difference between prokaryotic cells and eukaryotic cells. Again, we're going to be focusing mainly on eukaryotic cells for the purpose of this semester, which is what human cells are. But prokaryotic cells are, again, much more simple. They do not have a nucleus. They do not have internal membrane organelle systems. They don't have an intermembrane system. This includes things like eubacteria and archaea bacteria. Eukaryotic cells are going to have nuclei and multiple different sets of organelles that are going to be organized in a way that they're each going to have a specific function that they perform for the cell. That's going to include protists, fungi, plants, and then of course animals, which is where humans are going to fall into. Okay. Prokaryotic cells have a specialty nucleoid region that resembles the nucleus. It's going to have the DNA molecule, which in this case is going to be circular and singular, so much less complex than what we see in eukaryotes. And then it's also going to have the cytoplasm, which is going to have ribosomes. What ribosomes are going to be the site of protein synthesis. In a eukaryotic cell, ribosomes are separated from the DNA. So we're going to have the DNA copying into the RNA, and then the RNA is going to leave the nucleus. Then that RNA is going to be going out into the cytoplasm where it can meet up with the ribosomes to be able to make proteins. However, in the prokaryotic cells, it all kind of happens in the same place. There's no separation between DNA to RNA and RNA to protein. We do have a cell membrane, and we talked about that, phospholipids, cholesterol, multiple different proteins like channel proteins, right? We also are going to have a cell wall that's going to be found in certain things such as the bacteria, for example. Not all of them have this particular cell wall, but some of them do, and so we also can have a capsule. So the cell wall can be either gram-positive or gram-negative, determined by a particular stain. Basically, it's going to be, does it have sugars and amino acids, or is it going to be comprised mainly of peptidoglycans and polysaccharides? And then a capsule is going to be an extra layer or an additional layer that's going to be a protein layer that's going to encapsulate this cell and provide it an extra layer of protection. Prokaryotes also sometimes have specialty structures such as flagellum. The plural of them are going to be flagella. And we could have multiple flagella or just one. Typically, we have less of these than we do, for example, cilia. But flagella are going to serve in locomotion and can also serve in feeding the organism. We also can have what's called a pili, pilus, and the plural of that would be pili. And this is going to allow the bacteria to exchange information. They call them like a sex pili, basically. And they're going to exchange genetic information between bacterial cells. This allows one bacteria that has gained, for example, antibiotic resistance to share this novel genetic savior, right, this life-saving material with the other bacteria. So quite quickly in the population, we can end up with an antibiotic-resistant population. All right, so this is what a prokaryotic looks like. Again, prokaryotic cell structures are going to be pretty primitive compared to the eukaryotic cell structures. Here's a nucleoid region. That's going to be where the DNA is going to be kind of all round up. These are going to be ribosomes. Again, that's the site of protein synthesis, all housed in the cytoplasm. It's going to be surrounded by the plasma membrane and then the cell wall. And then here's the capsule. If we have one of those, the capsule is going to be, again, an additional layer. And this is a flagellum, which will allow this creature to be able to propel itself through a liquid medium. All right. Now, again, we talked about the plasma membrane, so I'll skip shortly over that. The cytoplasm, again, it's going to include the fluid portion, which is called the cytosol, as well as the membrane portion. The endomembrane system includes the nucleus, and then the organelles. So this is what's going to be found in the eukaryotic cells. Remember, we don't see organelles inside the prokaryotic cells. We also have a cytoskeleton and a nucleus. Remember, the nucleus is a specialty region that's a membrane-bound region that houses the DNA. It's kind of like the boss's office. It's going to direct everything that happens down on the factory floor, but nobody actually leaves the boss's office to walk down to the factory floor. They just send out a memo in the form of RNA, which then, of course, transcribes the protein actually over in the cytoplasm by the ribosomes. All right, so we also have a cell wall, which can be found in specialty types of eukaryotic cells like fungi and protists and plants, but we don't have that in animal cells, right? So plant cells are going to have that cell wall that allows it to have its rigid nature and what we call turgor pressure. So if we put fluid in there, it's going to help push back out, and that's going to help make the plant stand up. But we as animal cells, we don't have that cell wall. We're going to be more of a blobular shape than, say, a brick shape. Okay, so this here is the eukaryotic cell and all the organelles that we're going to discuss today. Don't worry, we're going to go over each of them independently. So even though I'm going to throw them all at you right now, you're going to get your chance to go over them in detail. We're going to start with the nucleus. The nucleus is going to be this big purple region here. Inside that we have the nucleolus, which is the site of ribosomal synthesis. We also are going to have outside here, this is loose chromatin, which is the DNA. But the DNA normally is going to be just completely unwound and accessible. But when we think of DNA, we think of it in chromosomes, which happens when we're undergoing cell division. But this cell is not undergoing cell division, so the DNA is going to be unwound again in what we call chromatin. And that means it's going to be accessible for the RNA to be able to make a copy, for it to be copied into RNA, let's be clear. So copy or the transcript of the RNA is going to be allowed out of the nucleus through what's called the nuclear pores. Those are depicted here like these little flower or donut-shaped rings. And when the RNA is allowed out, then it's going to be able to find its way to the ribosomes, which are shown here as these little dots. So we have free ribosomes and the ribosomes that are on the rough endoplasmic reticulum depicted here. Either way, we're going to have that. It's going to be the site where we're going to be assembling the protein out of our RNA transcript with the assistance of the ribosomes, right? Let's see, what else do we have here? So this is the nuclear membrane, right? So we have the nuclear pore complex and the nuclear envelope. I want you to take note that the nuclear envelope has 2 layers of plasma membrane. So it's not just one, it's going to be a 2 bilayers. But that's also going to connect directly, that nuclear membrane is going to connect directly to the ribosomal membrane. So let's move on, to the endoplasmic reticulum membrane where we're finding the ribosomes. Apologies. So let's talk about that endoplasmic reticulum. We have 2 different types. We have the rough endoplasmic reticulum, which is so named because it's studded by these ribosomes. So when they looked at it under a microscope, they just saw a bunch of rough dots. So they called it the rough ER because that's literally what they saw. And then this is the smooth ER over here. It does not have ribosomes, but it does play a role in the creation of lipids. It creates vesicles. So it's really important to the synthesis of lipids and other things for the cell. So even though it doesn't create protein like the rough ER, the smooth ER is still very important. So again, rough ER ribosomes, smooth ER no ribosomes. What else do we have to talk about here? These here are centrioles. We have a centriole pair that's going to be involved in the cell division. So right now it's not doing a lot because again the cell's not dividing, but when the cell's dividing we're going to end up with a centriole pair on either side of the cell, and the centrioles are going to have these little microfibers, spindle fibers, that are going to reach out and grab the DNA, which at that point will be condensed into chromosomes, will have lost the nucleus, so it's going to look totally different than this, and it's going to pull the chromosomes to either side during the process of cell division. We also have specialty things here called the mitochondria. This is going to be the site of ATP synthesis. You may have heard of the powerhouse of the cell or the battery of the cell. And we have the lysosome here, which is a cellular trash can. It's going to help break things down or digest things. We actually have specialty types of cellular trash cans like peroxisomes and proteasomes that are going to break down. You can consider them kind of like specialty recycling bins that break down particular types of discarded products. All right, so let's go back to the endomembrane system, which is what's going to happen directly after we're leaving the rough ER. So from the rough ER, proteins are going to be created, and again by these ribosomes, and then they're going to come off in little blebs. Those blebs are going to, now are depicted here in purple, are going to leave the rough ER and head over to this purple machinery here, which is called the Golgi apparatus or the Golgi complex. And just like a mail system is going to sort things as we go through into whether or not it's going east, west, north, south, what neighborhood, what region, etc., we're going to get more and more information as we're passing through each layer. So the vesicle is going to bind and then come across that and then come to this one and eventually get all the way out to here where it's going to be comprised mainly of everything that's headed to the same region, right? Just like the mailman's bag finally has letters that are all going to the same street. Then they're eventually going to leave off of this side of the Golgi and go wherever they're meant to be, whether that's embedded in this plasma membrane or headed over to the mitochondria or off to the lysosome for degradation or going back to the ER for some reason or whatever, wherever they're headed, they're going to come off of the other side of here with a little tag on it that basically gives it its little mail ID that says where it's going to go. All right, so I think I've touched on everything here in the eukaryotic cell. We will be focusing again mainly on the animal cell here, so you don't see any cell walls here. If you did see a plant cell, you would see a cell wall, and you would also see chloroplasts. All right, so that was kind of the rough overview of what I'm going to tell you. So now I'm actually going to tell you each particular organelle. So let's start with the nucleus. The nucleus is where we're going to have the DNA contained. It's housed here. Remember, we have 46 chromosomes in humans, but we're not going to see chromosomes all the time. We only see chromosomes during the process of cell division. Normally, we see chromatin, which is the uncondensed DNA that's going to be kind of sprawled all over and accessible to have RNA made from it. Surrounding this region, we're also going to have the nuclear envelope that's shown here in purple. Nuclear membrane is going to be a double membrane that makes that nuclear envelope. We have nuclear pores that are embedded in through this whole region. Again, they're depicted like little donuts. They're going to have little rings. They're going to have little gate guards there. And the gate guards are basically to ensure that the RNA can be allowed out, but that the DNA stays in place. So it's going to ask a couple of questions. First, it's going to ask, which sugar are you comprised of, right? If you're comprised of ribose, you might be allowed out. If you're comprised of deoxyribose, like DNA, you're not going to be allowed out. Are you single-stranded or double-stranded, right? DNA is double-stranded and it's not allowed out. RNA, single-stranded, you may pass, right? And then the last question is, are you going to have uracil, which is the nucleic acid that is going to be used by RNA only and not by DNA, right? If those three questions are answered correctly, then the RNA transcript is going to be allowed to exit out of these nuclear pores and find its way to the ribosomes, whether they're free ribosomes or on the rough ER, okay? We also have this nucleolus region here in the center. That's going to be the site of transcription of ribosomal RNA. Again, we're going to need a ton of ribosomes all the time for the creation of proteins because these are the little protein synthesis machineries. All right, from here, after the RNA makes it out of the nuclear pores, it's going to head over to the rough ER, right? The rough ER is studded with ribosomes and is the site of protein synthesis, which is called translation. The smooth ER is not going to have any ribosomes on it, and it's where we're going to be creating lipids and carbohydrates. All right, inside muscle cells, the smooth ER is also going to store calcium, and calcium release is going to be part of the activation of the muscle fibers. Okay, so after the protein's created on the rough ER, then it's going to shuttle over to the Golgi apparatus. Golgi bodies are going to be accepting little transport vesicles that, again, come off of the rough ER. And there's going to be a series of flattened stacks or membranes that are going to collect these transport vesicles and then modify the proteins and sort them and distribute them eventually into these little secretory vesicles, each of which is going to contain similar proteins that are headed to the similar location. Right, so as we go through here, we're going to end up with modifications that give more and more and more information about where this protein is going to end up. Some modifications are going to be in the form of added carbohydrates to form glycosylated proteins or glycoproteins. All right, mitochondria we described previously as the powerhouse of the cell or the battery of the cell. Mitochondria are specialty cells that were actually prokaryotic organisms engulfed, or at least the hypothesis is, the endosymbiotic hypothesis is that they were engulfed many moons ago in evolutionary history, engulfed by larger cells. And instead of the cell that engulfed them destroying them and eating them, they realized that the mitochondria has a specialty ability to be able to create ATP. It does this quite well through the process of oxidative phosphorylation. What's the difference between the ATP that's created in the mitochondria and the ATP created by the rest of the cell? Well, in the rest of the cell, we can only take a 6-carbon molecule and break it down into 2 3-carbon molecules. We can take those 3 carbon molecules and break them all the way down into carbon dioxide using oxygen, which we don't use in the cytosol. And in this oxidative phosphorylation process, we're going to generate 10 times the amount of ATP that we were generating by breaking down those 6 to 3. So this is going to give us 10 times the amount of energy, more bang for our buck for every sugar molecule that we started with. And that means that the organism that originally engulfed this mitochondria recognized its value and basically preserved it so that it could use it to be able to create and collect ATP. What's interesting is that they have 2 layers, an inner layer and an outer layer, and that inner membrane, these are called cristae, these folds, is where we're going to actually have our show, right? That's where we're going to be creating the energy. We're going to have the electron transport chain eventually producing ATP. What's also interesting is that this inner layer is going to be unique to the mitochondria. It's going to have its own mitochondrial information. It has mitochondrial DNA inside here that's unique and separate from the rest of the cell. But the outside here, this outer membrane, is going to mimic whatever cell line it's in. If it's in a dog, it's going to be a dog membrane. If it's in a human, it's going to be a human membrane. So that outer layer is going to mimic whatever it is to replicate self, basically, so that it can hide underneath the radar and create the ATP. It's called symbiosis when both organisms get something out of the deal. So the mitochondria get protection and the cell gets the generation of ATP. So it plays a really important role in cell cycle. It's also going to be involved in cellular aging and cellular death. So mitochondria are really important in a lot of different stages of what's going on with the cell. Okay, so let's talk about ribosomes for a second. Ribosomes are found either free in solution or bound to the rough ER. And they're going to be the site of protein synthesis or protein translation. They're not actually a membrane-bound organelle, but they're right next to the rough ER usually. And they're comprised of ribosomal RNA. We have a large subunit and a small subunit, and it's kind of like a zipper. They're going to lay on either side of the mRNA transcript and shuttle along this transcript, reading it and turning it into protein. Okay. We'll talk about that in a little while, perhaps in a different chapter. We're also talking about here some of the, I called them cellular trash cans earlier. They're also perhaps cellular recycling bins. These are going to be parts of the cell where we're going to take products that we're not able to use or degraded or misfolded or something that is even brought in because we've digested it from another organism and it's whatever the byproducts are that are left over as trash, but we're going to want to destroy, right? So we have specialty digestive enzymes that are found in lysosomes that break down the macromolecules in old organelles and again, anything that we would consider to be trash. And we also have specialty cells like immune cells that have a ton of these lysosomes because immune cells serve to encounter and digest foreign pathogens. And so they're going to have a lot of excess trash to degrade, right? And then peroxisomes are going to be specialty cells that specialty regions in the cell, organelles, that break down fatty acids. And they do that by using peroxide, right? So they use peroxide to help detoxify certain substances. These are found in high amounts in the liver because the liver is responsible for detoxifying the blood. Okay, centrioles are specialty organelles that are going to have spindle fibers. Spindle fibers are microtubules that are going to help transport the chromosomes to either side of the cell during cell division. They're going to be found in triplets. We have 1, 2, 3, 4, 5, 6, 7, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yes, so we have 9 of them. 9 of these triplets, and they're going to run alongside and encounter the chromosomes. They'll connect at the center of the chromosomes. At this point, the chromosomes are condensed, of course, into chromosomes, into little X's. And they'll connect at the middle with a region called the kinetochore, which is at the center of the centromere, when allowing the sister chromatids to be decoupled, having one set going to each side of the cell. So again, they're involved in transporting the chromosomes during cell division. These are different types of cytoskeletal proteins or structural proteins that are involved in the cytoskeleton. We have small filaments like actin. We have microtubules, which are round into a kind of like a spring. And then we have intermediate filaments as well. And all of these are going to be found connecting different organelles to one another. So again, they're not just free floating inside here. They're all going to be connected through this scaffolding network. And the interesting thing about the scaffolding network is it doesn't just serve as connective material. It also serves as kind of like a meeting place. So oftentimes things that come in from the outside of the cell will be relayed to the inside of the cell via things that are connected at these scaffolding proteins. Okay, so again, microtubules are going to be hollow tubes, and they're going to be what spindle fibers are comprised of. We're going to use spindle fibers during mitosis and meiosis when we're separating chromosomes from one side or the other of the cell. They also are going to be in the middle of the flagella of eukaryotic sperm cells, and are also going to be used as pathways or walkways for motor proteins to be able to transport things from one part of the cell to the other. So they're kind of like catwalks as well as the structural proteins. All right. Intermediate filaments are going to be about 10 nanometers in size. They're intertwined as a rope, and their function is structural. Again, they're acting as a scaffolding, keeping the organelles in place, holding the cell shape in place. We also have microfilaments, which are going to be a little bit smaller. These are about 10 nanometers in width. They're generally composed mainly of actin. And these guys function in crawling motion, so when the cells are going to move throughout the body, for example, immune cells migrate towards an infected site because they have gotten a signal that there's something that they need to go towards to be able to digest, or cellular damage so that we can hold things together while we're having the repair mechanism. All of this is going to be done by microfilaments. It's also going to be it's also involved in cytokinesis, which is when we are forming a cleavage furrow between two separate cells. We're basically taking an O or a long oval and we're turning it into a figure 8 and then into 2 lowercase o's, right? We're separating one cell into 2 cells during cytokinesis. These are some examples of extracellular proteins like collagen and elastin. These are going to give tissues strength and flexibility. So collagen is going to be strong. It's going to resist in stretching. It's going to be found in things like your skin, your bones, your tendons. Elastin is going to give the flexibility to the skin, tissues, bladders, lung, etc., blood vessels. So elastin gives flexibility. Integrins are going to be proteins that span the entire membrane that send informations from the outside of the cell to the inside of the cell. Fibronectins are connecting proteins. They connect integrins to extracellular proteins. And we also have glycoproteins such as proteoglycans that are going to hold water and allow hydration in places like skin and joints. So these are just examples of different types of extracellular proteins. Again, proteoglycans are going to hold water, collagen going to be very strong, elastin going to be very flexible, integrin connects to fibronectin is going to be in purple, connecting to integrin, which is a transmembrane protein, which is connected to the actin on the inside. So integrins are going to connect intracellular proteins to extracellular proteins and become basically adhesions. So extracellular matrix proteins are very important for connecting cells both to each other and to the base of the membrane. It gives them strength and flexibility. And depending on which tissue you look at, you're going to see different amounts and also variability in the types of the extracellular proteins that are found in those regions. Okay, so let's talk about junction proteins. We have cell junction proteins. We have 3 different types, including tight junctions, adherent junctions, and gap junctions. These are specialty types of proteins that connect 2 cells to one another and allow for communication amongst cells. Tight junctions are going to help hold cells together and allow, this is going to be a waterproof barrier, so it's going to prevent molecules from moving between these cells. I think it's going to, example, this is going to be like the blood-brain barrier. We also have adhering junctions. These are actually going to anchor cells instead of to one another. They're going to anchor them to the extracellular matrix. And these are going to be really important for cells that undergo a lot of stretching and need to be able to hold firm, like skin, for example. Need to be very resilient. And then gap junctions are going to be involved in communication. These guys form channels that directly connect cells together and allow them to communicate to one another. A good example of this is the heart muscle, allowing for simultaneous contraction along the entire muscle. All of the cells have to be acting in concert. All right, so here we have some examples here. Here's a gap junction. These are connexin, which is going to be protein that's going to allow us to make this particular gap. You can see this looks like an old school telephone perhaps, right? Information can go straight on through from one cell to the other. So instead of information having to go from outside the cell into the extracellular matrix, come over and then come back in, it can be relayed directly across so that we can kind of coordinate things like, for example, a heart cell contraction, right? Tight junctions, I mentioned, they're going to be kind of like, they are waterproof. They're going to be kind of like a Ziploc or a zipper, but they're going to be like this. Going to have a bunch of proteins called occludin that are going to be holding them separate. Again, this is going to form a watertight seal in places like your bladder, etc. And anchoring junctions, again, they're going to be connecting to the extracellular matrix. In this case, they're cadherin proteins they're connecting to, and that's going to go across the intermediate filaments on the inside of the cell. So anchoring junctions are going to connect cells together by way of connecting them to the extracellular matrix. Okay, so here's an application that we're going to discuss at the end. So if John's going to cycle on his bike, and it takes him about 3 hours to complete this ride, and he's going to sweat approximately 1 liter, which drink would be best for rehydration and why? A drink that is hypertonic, isotonic, or hypotonic. And we haven't really talked about this yet, but that's basically going to mean what is the concentration of salts and water relative to the concentration of salt and water inside the body of the individual. Okay, and it's asking you specifically to focus on something like a Gatorade or a sports drink that's going to contain electrolytes or specialty minerals that are going to help replace things that are lost in the sweat. Okay, and we'll get back to that towards the end of the class. Okay, so as I mentioned previously, a plasma membrane is what we call semi-permeable. Some things are going to be allowed through and others are not, right? And that's because we have a specialty barrier function made up by phospholipids because of the fact that these phospholipids are amphipathic. What does amphipathic mean? So when I showed you previously, you saw a region that interacts well with water and a region that interacts well with oil, right? A hydrophobic region and a hydrophilic region. And that's what's called amphipathic, a molecule that has both regions. So it can interact both with things that are oil and also with things that are water-based. And so certain molecules are able to go across this membrane, like water and small things, ions. But large molecules, particularly charged molecules, have to cross with permission. Again, they get this permission from membrane proteins, basically. It's going to be highly regulated, and it's only going to be, the cell's only going to allow in what it wants to bring in and keep out what it wants to keep out. Things can come into the cell from a multitude of ways. They can come in from simple diffusion, which does not require energy. This is basically when molecules are moving from a high concentration to a low concentration directly across the membrane. Small things are able to do this without the assistance. Again, there's not going to be any assistance from the membrane protein required. This is going to include things like oxygen, carbon dioxide, nitrogen, short-chain fatty acids, glycerol, and water. Simple diffusion here is just shown as this is not diffusion across a membrane. This is just shown diffusion in water. But if you were to start with a drop of dye and water over time, you would end up with all of the water being the same color of blue, right? Because eventually as the water molecules knock into one another, the dye starts to diffuse out and eventually reaches a point of equilibrium. What do I mean by that? When the entire solution has the same concentration. Okay, so when we're looking at the extracellular fluid and the intracellular fluid, we are always going to have a gradient. That means a difference in concentration between the outside and the inside. And the cell maintains that gradient in a lot of different ways, like pumps, etc. But that means that it's able to bring in what it wants and keep out what it doesn't want. And the way that we're going to come in by simple diffusion is going to be from a high concentration to a low concentration with the gradient across the membrane without the assistance of a membrane molecule. You have to have a small molecule, something like oxygen, etc. And again, it's going to diffuse from high concentration to low concentration, right? See, on this side we have 7, on this side we have 3. Diffusion is going to be affected by many different things. The concentration gradient, so let me go backwards. If we had 20 on this side and only 3, it would move much faster if we had 4 on this side and only 3, right? So the difference in the concentration on one side and the other is the intensity of the gradient. And the concentration gradient affects the rate of diffusion, as does the temperature. Increasing the temperature is going to increase it because things are going to be bouncing off of each other and moving much more quickly. The surface area, the diffusion distance, and the size of the molecule are all also going to affect the rate of diffusion. Now let's talk about the diffusion of water. That's called osmosis. Again, we're moving from levels of high water to low water, so from high concentration to low. We're also not going to require any ATP. And we can go through the process of simple diffusion, but we can also cross cell membranes through specialty proteins called aquaporins, which are basically going to be membrane channels that allow water to cross. Remember, water is a polar molecule. It means it has one side that's going to be a little bit slightly positively charged and a region that's going to be slightly negatively charged. But even though it is a polar molecule, it's small enough that it goes right across the membrane without a membrane protein necessary. So it can diffuse across the water, across the membrane without the need of specialty proteins, although it also can be assisted by aquaporins that are specialty proteins. And when we're talking about the motions of water across the membrane, that's called osmotic pressure. The concentration of solute relative to water on either side of the membrane is going to affect the ability or the propensity of water to travel from one direction on the membrane to the other. Okay, so for us to understand what's headed on next, when we were talking about isotonic, hypertonic, and hypotonic and that application question, in order for us to understand that, we need to understand what is a solute, what is a solvent, how do they work. Okay, so a solute is going to be any chemical that's going to be going into the liquid solution. A solvent is going to be the liquid that accepts that chemical. So if, for example, you're putting 2 tablespoons of salt into a glass of water, salt or sodium chloride would be the solute, water would be the solvent, and water is considered the universal solvent. Now we're going to be looking at the concentration of solute in solution based on the concentration of the solute on the outside relative to the solute on the inside. And we measure osmotic concentration in milliosmol. So the cell typically is going to have approximately 30 milliosmol. And that's going to be also measured in another way, 0.9% sodium chloride. This is the normal saline concentration. So again, we can look at molarity or we can look at concentration. Either of these, or like percentage concentration, are going to be just descriptions of the concentration of solute in solution. Now, when we are looking at the solutions relative to other solutions, that's when we start using the words hypertonic, isotonic, and hypotonic. So let's start with isotonic because it's probably the easiest to understand, right? If we have the same concentration in one liquid on the outside of the cell as we have in the liquid on the inside of the cell, that's called isotonic. In this case, water will move freely about from one side of the membrane to the other, and it will be approximately equal in concentration, and the cell will stay the same size. Now let's look at the word hypertonic. Hypertonic solution is going to have more of a solute than the cell does. That means that it has less of a water. Okay, so if you look at the more solute, let's look at solute concentration as 95%, water concentration as 5%. That's what, right? So if we have 95 and 5, of the solute concentration, and then we're going to be up against a concentration, a fluid that's going to have, say, 98 and 2%. So it'll have 98% water and 2%. That's going to be considered, 98% solute and 2% water. Gosh, I wish I could draw for you guys. That's going to be considered hypertonic because it has a higher concentration of solutes than the cell. And remember, the solutes can't go from one side to the other. Only the water can. So now instead of looking at the higher solute concentration, remember, a hypertonic solution is the one that has 98% solutes, and the hypotonic has 95% solute. But let's look at the water, right? If we have 98 over here and 95 here, that means we have 2% water and 5% water. And water is going to go from the higher concentration to the lower concentration. That means water is going to leave the hypotonic solution, right? Leave the hypertonic solution and go to the hypotonic solution. So in the hypertonic solution, the water will move from the cell into the solution and the cell will shrink. It will shrivel up. If we put the cell into a solution and say it's 100% water, so it's for sure hypotonic because it's going to have way less solute, it has 0% solute than the cell itself. Water is going to move from that 100% to, say, what is it, with 95% inside the cell. So it's going to be 100% water and only 95% is going to move inward. So in a hypotonic solution, it has less solute than the cell. Liquid's going to move from the solution into the cell. The cell is going to swell up and could burst open. All right, this diagram should hopefully help clarify what I was just saying. So here we have a cell in isotonic solution, right? It's described as isotonic, and you can see we have 5 inside and 5 outside. That means that we have the same amount of solutes on the outside as we do inside. Water is going to be moving in and out at the same amount, and the cell is going to stay the same size. If, however, here we have a cell being put in a hypotonic solution. In this case, the hypotonic solution is going to be all water, right? It's going to have less salt than the cell. Remember, the salt can't leave. So in order for us to try to make it equal, the water is going to have to move from here it's at 100%, here it's only at 95%. So the water is going to have to move from 100 to 95%, causing this cell to swell up and eventually to burst. If, however, we put the cell in a high salt concentration, so there's more salt on the outside than on the inside, the water inside the cell will have the tendency to move outside the cell to try to match the concentration of salinity outside the cell, and the cell will shrivel up. Okay? All right, so that's all talking about diffusion and osmosis. That was simple diffusion and osmosis. Let's talk about facilitated diffusion. As I mentioned, there are specialty membrane proteins that are embedded all the way through the membrane that allow particular things, whatever they are, substances, molecules, to go across this membrane. These molecules are going to be moving down their concentration gradient, so from high concentration to low concentration. And because of that, we're not going to require energy. We're only going to require ATP when we're moving across a concentration gradient. So when we're moving down a concentration gradient, again, it's going to require no energy at all. And we've got these specialty types of carriers. We have channels and carrier proteins. Channel proteins are going to allow ions to cross, and once it's open, it's basically like an open door, anything can flow in. Carriers, on the other hand, are going to move one thing at a time, and therefore can become saturated, so they have a rate at which they're able to move things across. So you can picture this more along the lines of, say, an elevator than an open door, right? Only a certain amount can get in there, then they have to move, and then it has to come back. So this is going to result in the fact that carriers are going to be able to be saturated, whereas channels are not. Here we have facilitated diffusion of glucose using a specialty carrier or channel protein. All right. Here we have glucose coming in. It's going to be bound here. It's going to then go into the cell by way of a conversion of this molecule, this carrier molecule. And just as I mentioned previously, there's more glucose on the outside of the cell than the inside of the cell, at least depicted in this image. So we're bringing glucose in. So we're going with the gradients. Again, from high to low, so no energy is required. So facilitated diffusion is really important in the maintenance of what we call homeostasis. Remember, we have a ton of different ions that we have to maintain at particular concentrations inside the cell, no matter what's going on outside of the cell. So we have to have a specific balance inside and out, and the concentration of things like sodium and chloride, etc., is going to be different than other things such as potassium, etc. So concentration of sodium and chloride is higher on the outside, potassium is higher on the inside, calcium is higher inside the smooth ER inside the cell. So different ions are sequestered at different concentrations inside v outside the cell, and that's kind of depicted here, right? Higher outside, lower inside, higher inside, lower outside, higher inside the ER of the cell. So that's what this image here is trying to depict. Okay, so all of the methods of transport that we just previously talked about do not require energy. No ATP needs to be utilized in order for this to happen. Now, we also have a type of transport called primary active transport, which is going to allow cells to transport things in against a concentration gradient. This means we're going to have to require ATP or energy. Picture that you're sitting in a boat on the water and there's a hole in the boat. Letting the water in from the higher amount of water on the outside to the less amount of water inside the boat, easy peasy, right? Bailing the water out to make sure that you keep less water inside the boat than outside in the lake takes energy, right? And that's active transport, bringing things in against a concentration gradient. And we do this by a couple different ways, but one of the primary ways that we do this is a series of pumps. One of the main pumps we use is called the sodium-potassium pump. It's going to utilize energy in order to pump 3 calcium out of the cell and 2 potassium into the cell. In this way, I just said sodium. I just said calcium. I'm sorry, I meant sodium. 3 sodium go out and 2 potassium come back in. In this way, we're able to make sure that we keep what I just mentioned here, more potassium in than out and more sodium out than in. Okay, so the sodium potassium pump is going to be a really large energy expenditure. Almost a third of the energy expended by the cells is used to drive these membrane proteins, such that we can keep this electrical gradient and a chemical gradient, so that we have a concentration gradient of particular things like sodium and potassium. And we also are going to have a electrical gradient where we have a more negative relatively more negative inside of the cell and a relatively slightly positive extracellular fluid, right? And this is really important, especially in certain cells that require electrical stimulation, like nervous tissue or muscle tissue. All right, so here's an example of the sodium-potassium pump. Here we're going to have 3 sodiums lining up. ATP is going to come in. ATP is the high energy form. Remember, triphosphate is high energy. When we go from triphosphate to diphosphate, we're going to lose a phosphate, but that's going to convert the sodium. So we have a conformational change in the carrier, making it so that the sodium can go to the outside, and the potassium is going to pop right in here. When that happens, the potassium is going to come back for free, basically, and the phosphate is going to be released as a free phosphate that can then be bound back to an ADP molecule in the presence of some other energy creating process. So when energy is released, that will drive the creation of ATP again. Again, this is going to be an energy intensive process because ATP is broken down here and released to be able to force that sodium out such that potassium can bind. All right, that's a very good example of active transport. Another good example of active transport is the proton pump. Remember, we just talked about the fact that we need electrical gradients on the outside via the inside of the cell. The proton pump is going to utilize energy to pump protons across the membrane. This is particularly prominent in mitochondria, where this gradient is going to build up inside the intermembrane space and is going to be utilized for the electron transport chain, right? It's going to give us a concentration gradient, allowing the re-entry of protons back into the cell. And when we utilize that, then we can generate ATP. As I mentioned, ATP is created when we have the diphosphate and the free phosphate in the presence of some sort of energy. And in this case, the protons coming back in are going to create energy that are going to drive the creation of ATP. That's called chemiosmosis, and it's going to be one of the primary ways that we get energy to the cell. Again, in the mitochondria, it's going to be very essential in the process of energy metabolism. Right? So again, here's the proton pump. We're going to have protons going in, and in the process of sending the protons in, we create a gradient. That means we have more protons outside here in the intermembrane space than we do in this matrix. That means that protons are going to free flow back across, right, from high to low. When they go from high to low, we're going to also have ADP waiting in the wings with free phosphate, and the energy that's created when that hydrogen ion comes in, that proton comes in, is going to drive the synthesis of ATP from ADP and free phosphate, because we have ATP synthase, which is an enzyme sitting right here as well, which again converts ADP into ATP, when we have this hydrogen ion coming across this proton pump. Okay, so proton pumps are membrane proteins. They're found in the inner mitochondrial membrane, and they're vital for the production of ATP. All right, so I'm going to switch gears for a second here and talk about coupled transports. Coupled transport happens when we move two things across a membrane at the same time. And this can happen when we're moving two things in, or when we're moving two things out, or when we're moving one thing in and one thing out. If both things are moving in the same direction, that's called symports. And if they're moving in opposite directions, that's called antiport, like that sodium-potassium pump that we just talked about, right? And in this way, we can actually kind of bring things in across their concentration gradients in the wings of something else. So if, for example, the cell wants to bring in molecule A and molecule B is going to hitch a ride, but the cell really needs molecule A, so it's going to allow molecule B in anyway, even though it's not necessarily normally going to allow molecule B in. Or vice versa, we want molecule A in, and in order for molecule A to come in, molecule B has to leave. Think of this pretty girl at the club who says, I'll go in only if my ex-boyfriend gets kicked out. So then the bouncer goes and asks the ex-boyfriend to leave so that she can come in. That's the antiport, right? One molecule goes one direction, the other molecule goes the other direction, like the sodium-potassium pump, right? And we also have secondary active transport when we're going to be using the sodium gradient, as I just talked about. So we have a higher amount of sodium outside the cells, and that's going to pull other nutrients like glucose and amino acids into the cell so that ATP is not required. So let me go backwards for a second here. The fact that we have more sodium on the outside than on the inside means that sodium is going to flow in freely. That's why we have to constantly pump it out, right? Here's this pump, right? Because we always have a lot more inside than outside. Have to pump it out, it's going to flow back in. So if when it flows back in, it flows back in with something that is in a lower concentration, like, for example, sugars, right? So again, we have the sodium being pumped out, the potassium being pumped back in, and when the sodium free flows back in, it brings the sugar with it. That's coupled transport or secondary active transport. Basically, we have the simultaneous movement of two molecules through the same membrane protein, whether in the same direction or in opposite directions. If they're moving in the same direction, that's symport. Opposite directions is antiport. But large molecules aren't able to just move across the membrane at will. Bulky substances are going to need to go by vesicular transport. Basically, they're going to be contained within a vesicular membrane, and they're going to be moved inside and out of the cell when that membrane touches the cell membrane and opens up. So endocytosis is when the cell membrane is going to engulf substances that are outside of the cell and bring them inside of the cell by forming a vesicle that's then going to end up inside the cell. We have two different ways of this. We have phagocytosis, which is how solids are going to be brought in. And this is how an immune cell, for example, is going to digest a pathogen like a microbe or a bacterial cell. We also have what's called cell sipping or pinocytosis, where fluids are brought in, where small amounts of fluids and nutrients are going to be brought into the cell. An example of this is when the human egg, what the human egg does just prior to fertilization, where it engulfs a bunch of fluids and stores nutrients for the in hopes of the developing embryo. Exocytosis is the opposite of endocytosis. When substances are discharged from the cell in the form of vesicular transport, basically the vesicles are going to touch the inner surface of the cell, bleb off, and release whatever their product is. So again, here's going to be what we have when we are pulling in things like large things like microbes, etc. It's going to be called phagocytosis here, pinocytosis here, and this is exocytosis when things are leaving. All right, so two different types of endocytosis. Large molecules brought in by, again, phagocytosis. Small molecules is by cell sipping, so they bind to the membrane, and they pull in and make an invagination or a dimple, and eventually form a vesicle. And then exocytosis when things are exiting the cell, when they're going to be coming up in secretory vesicles that touch the cell membrane and join with the cell membrane, and the secretory product is released in the extracellular fluid. Thank you guys so much for taking the time to talk with me today. I guess I should say that the answer to that application was to drink an energy drink so that you could make sure that you're restoring all of your electrolytes. All right, thank you so much for joining us today. We will see you next time for Unit 2B. Have a fantastic day and happy studying.